Okay, so I just want to show you some cool stuff I found today. So this is ZRSR2. To remind you, ZRSR2 is the protein that's involved in finding the 3' end of U12 introns. It's also called URP, URP, or ERP, I guess. And it's a homolog of U2AF35, which Brad's been working on and which is responsible for finding the 3' end of U2 introns. The story, as I've been discussing with Graham and Brad, is even actually a little more complicated than that, but let's stick to one issue right here, which is alternative splicing. So this has been looked at before. So here we're looking at flies. So this is one of the two twintrons in flies. So you can see either this boundary and that boundary gets used, or this boundary and that boundary gets used. So to remind you, these thick bars mean that that's protein coding sequence. So what that means is we start over here, we got protein coding sequence, protein coding sequence, protein coding, sorry, let's start on the bottom one. Protein coding sequence, intron, protein coding sequence, intron, protein coding sequence, intron, protein coding sequence, all the way to here. So that's if we use these two boundaries. Sorry. If we use these boundaries, however, we see we get protein coding sequence, but then we hit the fact that this stops being thick means that it's the end of the region with protein coding potential, which means it has a stop codon. And so actually we can see that right here. So let's blow this up. So we want to look at these two boundaries and those two boundaries. If we want to look at sequences, we'll look at one at a time. So let's start with this pair. So that pair there is this pair here. And so again, we see these fat lines are the coding sequence, and you can see that this fat line stops right at the stop codon. Okay, so that means that this is a stop codon, that's a stop codon. So that means if we use this pair of boundaries, we hit a premature stop codon and probably don't make protein. Probably the RNA is degraded. Okay, so let's look at what these boundaries are. This first one, so this is the first splice boundary, that's GT8CGT. So that's not a U12, that's a U2. But then here we see GT8CCTT, so that's a U2 boundary. Okay, so now we can look at the other side, the paired boundaries. And so down here what we see is, so for the one that's, so this is the U12, the bottom one is the U2, so it pairs with this one, and we can see that it's just an AG. And if we look down there, we don't see a clear U12 branch point. But then this top one then pairs with this top one, and so we see we've got our GT AG boundary. And then if we look up here, we see the classic CCTTAAC U12 branch point. Okay, so this is the U12 twintron that was reported by Lin et al. That we've talked about briefly. Oops. So today I started poking around a little bit more. This is humans. I'm hopefully going to remember to come back to it. Humans look the most different from what we're seeing. But in case I forget to come back to it, let's just do this quickly. So here what we're seeing, again, we have two isoforms. On the bottom, this is the long protein coding one. And then on the top, we have this exon that when it's included, it leads to a premature stop codon. So this is shown slightly differently. In the other case, this would have been shown joined to all this stuff. But in this case, it's shown just to the end, basically, of the protein coding sequence. And so then if we come down here to those boundaries, so this is these boundaries. Oops. Where the heck am I? Well, apparently I've lost my mind on this one. Yep, I totally grabbed the wrong bit. Oh, no, I didn't grab the wrong bit. Okay, so here, in fact, if we looked at these boundaries, what we'd see is that this is also a U2. So this is U2 and that's U2. What's going on here is that this exon is being caused to be used by some sequence that we're about to look at. So this has been reported by Freelander et al. And a couple of other genes. That basically you have binding of the U11 SNRP in this region, and that triggers inclusion of this exon even though it's a U2 boundary. So I can point you to those papers. That'll be a better way to look at it. Here, let me just show you. that we can see. Here's this GTATC TT, and then there's another one down here, ATTT I think is the one. In any case, I think that this is a candidate for a binding site for the U11, triggering inclusion of this exon and thereby truncating things. So basically what happens here is if you have an active U11 SNRP, it's going to bind here and trigger use of this exon, which is going to lead to this alternative transcript that doesn't make a protein. Okay, so that was not the best nor the simplest part of this presentation. So if you're not quite on board on that one yet, the rest of the stuff should be clearer. Okay, so this is sea urchins. Sea urchins are deuterostomes like ourselves, but are not chordates. So fairly distant from us, but not super distant. So this is a different, and you can see kind of inferior, genome browser, but that's what we've got it on. On the top here, we've got the predicted protein coding region, and then the transcriptome. So the transcriptome here is based on RNA-seq data that they actually assembled and then saw what they see. So one thing that they see, this isn't the whole gene, it's just the bit that I wanted to focus on. One thing they see is the predicted, right? This whole exon joined to this whole exon. And then this other thing that they see is this so they see this transcript, which differs in two ways. One is that this is a U12. One is, this pair of boundaries is used, and one is that this pair of boundaries is used. And so let's zoom in on this pair of boundaries. So this boundary is that boundary, this boundary is that boundary, and what we see if we look at it is AT-CC. So that looks like a U12 5'. Again, this one just looks like a U2. And then if we look at the other boundary, so here's the bottom boundary, and what we can see is that we've got to go with our AT-ATCC boundary, we've got an AC boundary, and if we look upstream, we see the CC-TT-AACC. And so what that means is now here in the same gene, in a different species, we have evolved a different system where now we've got inclusion of this extra exon. And actually, I forgot to include it, but if you look at these two boundaries, they look like they might be U12s as well. So instead of using this pair of U2 boundaries that gives you a protein, you get triggering of these U12s, which I haven't shown it here, but let's see, can we get it quickly? I don't think we can get it quickly here, but you can see that actually this thing right here, this stop codon is in frame. So if you get inclusion of this exon, that's that exon, you get an in-frame stop codon, so you disrupt protein usage. Protein production. So the top one makes a protein, the bottom one's probably degraded. Okay, so a second way to do it, or a third one if you count the human one I didn't do a very good job of explaining. This is Rhizophagus. This is Graham's favorite fungus. On the top here, again, we've got the protein coding form, and then we've got two down here that are supported by RNAs. And so again, this is the protein coding form, and again, if we look at this other form, we see we've got inclusion of this extra exon, use of this different boundary, and then hitting this stop immediately. So again, we get truncation of the form. And here it is. So here, what we're looking at is this is the extra exon that's included, and then this is the atypical boundary. So this little red nub here is this little nub there, and then this exon is that exon. Okay, so this is the alternative exon, and this is the alternative boundary. And so what you see if you look at these boundaries is they're actually kind of weird. And so I actually went and looked at the RNA-seq data. And so it's annotated at this AT boundary, but it's not an AT-ATCC, right? Actually, we can see that. This is the boundary that looks like the RT-ATC-YT. So, and if you look in the RNA-seq data, what you see is that this boundary is used, and then actually this AT is used, so that the splicing event is occurring from there to there, again giving you this in-frame stop codon, so yet another way to degrade things. And so far this looks like this might just be an evolutionarily conserved event, but let me tell you that if you look in other, well, for one thing, remember they concluded that it was new in Drosophila, so that is not conserved. We don't find it in humans. So it looks like again and again this thing has evolved this sort of similar kind of situation where we get inclusion of an exon, leading to that basically when you use U12 boundaries, you degrade the transcript. And here's the last one. So this is a Arabidopsis. You can't quite be confident here, but basically what we're looking at is two boundaries. This is the protein coding form, and then this is a cDNA that's actually observed. That was in an EST sequencing, I think. They randomly, shotgun sequencing, they randomly found this sequence. So if we look in at the sequence, so what I've done here, the red is, basically the capitals and lowercase represent the protein coding forms. So the protein coding form, this blue form. So what you can see on the top, kind of obscured, is some lowercase. So that's a short intron, then a short exon, then a short intron, and then a short exon, and then this long intron, all this red sequence. Okay, so I've highlighted in red this long intron. And then in boxes, I've included what is used as introns on these boundaries, right? So we've got this extra exon is actually this stuff in between here, between the asterisks. And then we've got one intron and another intron. So instead of using this 5' boundary, we use this 5 boundary, which says, you can see GT, AT, CCT, and then we use this extra C terminus, and we can see this, instead of, it's CTTTAAC, which is the branch point. And then, so all this stuff gets used as an exon, and then we use this new U2 boundary, and actually we use the old U2 3' boundary. So basically, there's this new usage of boundaries. Again, I haven't checked all of these, but I would bet good money that these U12 forms are going to lead to degradation. Yeah, so I don't think I'll go back to the human. We can talk about that some other time. It's a little bit less clear. I think actually we should just read a paper and then go back to it. But what we see basically, so let's just flip through these quickly. So in Drosophila we have a U12 boundary and a downstream U12 boundary, right? So U2. So we have downstream, so that's one form. In humans we have this triggering of a U2 boundary. In sea urchins, we have these 4 U12 boundaries, 1, 2, 3, 4 that get used, and notice that they're inside the U2 boundaries. In Rhizophagus, we use the upstream U2 and use a new U2, and then we have a new U12 downstream from that. We have a new U12 and a new U12. And then in Arabidopsis, here we have a downstream U12 and then a U2. So basically, and then we use the same U2 boundary. So basically, in these 5 different species, we're finding 5 different ways in which we have a kind of complex twintron situation, where if we use the U12 boundaries, we get degradation. If we use the U2, we get protein production. So all of these would be ways that if you have an active U12 spliceosome, you would thwart production of more of this U12 spliceosomal protein, which I think is real cool.